The next section, chapter 2, we'll talk about the organic molecules of cells. So most of the macromolecules that you'll be familiar with, we have the simple carbohydrates. We'll talk about the structures found therein, different types of ring found in carbohydrates. We'll talk about different types of lipids, in particular fatty acids, phospholipids, and steroids, how each of the composition will differ greatly and therefore also will determine the particular function of those particular lipids. We'll talk about the differences and similarities between RNA and DNA. We'll talk about the different types of amino acids, the polar, nonpolar, the bases, and the acids. And finally, we'll talk about how proteins fold in different structures and how different types of bonding, in particular the hydrophobic or hydrophilic reactions, other types of ionic bonds, even disulfide bonds, will basically configure how well a particular protein will fold and therefore function thereafter. We take our first look at carbohydrates. We look at glucose, since this will be the molecule we'll be dealing with mostly in most cellular types of reactions. So it shows here glucose in the linear form and also both of its ring forms, both in the alpha and beta. So that's important in particular when we start looking at how the individual monosaccharides will actually form linkages and how it will be oriented in the cell. We'll talk about different types of macromolecules, including the chitin. They'll look at the starches, the glycogen, as well as different types of fibers or cellulose. All has to deal with that one carbon on the right end there. That's the first carbon there. So depending on where the hydroxide molecule will lay, will determine how well these will interact with other forms of glucose. But very simply, you're looking at a 6-carbon glucose. Its molecular formula is C6H12O6. So that's your basic formula through there, and most of our polysaccharides are going to be derived on some kind of monomer of the C6H12O6 template. Now we link the different monosaccharides through what's known as a glycosidic bond. These are basically covalent bonds, usually dealing with a dehydrated synthesis type of reaction, and oftentimes we'll see the glucose will be isomerized or will be phosphorylized with different types of reactions. But anytime that we see a glycosidic bond, this is going to require energy because we're building a macromolecule. So this is going to be known as an endergonic reaction. And we look at different combinations of the different monomers that we see here. So in this case, we see 2 monosaccharides going to make a disaccharide through dehydration synthesis. So this is very simply a covalent bond, but because we're dealing with the particular macromolecules here, this is known as a glycosidic bond. So you're looking at 2 monosaccharides coming together to form a disaccharide through dehydrated synthesis. Now this is the end result here. We'll look in the next slide at the individual steps. Now ATP is needed to drive the initial segment of this particular reaction. So you have a glucose molecule here, which is going to accept a phosphate ion from ATP to the 6th carbon, and it'll change it into the glucose 6-phosphate. This will isomerize into glucose 1-phosphate, and then a UTP will come along and donate another phosphate with it to create another intermediate called UDP glucose. Now, this is going to be now an exergonic reaction, so it's going to release the UDP, and that release of free energy will allow the glycosidic bond between this glucose molecule and the next adjacent molecule in the 1-carbon 4-carbon linkage. And as you can see here, because it's in the downward position, this is going to be in the alpha linkage in particular, so most likely this is going to form glycogen. I just want to point out here too, with the glucose molecule, see atop of this diagram here. So basically, this is going to be an uphill style reaction. We have to have ATP input or free energy input to drive this particular type of reaction. So you can see that the 6th carbon atom is phosphorylated, and then it will isomerize into glucose 1-phosphate. So it's getting ready to accept the next phosphate to be activated again. So this now creates a downhill reaction to basically form the glycosidic linkage between it and also the next glucose. So we start chaining different monosaccharides together. We get different types of structures, each with its own different purpose. If they're small chains, they're known as oligosaccharides. If they're long chains, they tend to be known as polysaccharides. And the polysaccharides, depending on whether they're branched or unbranched, can have different functions. So in this case, we see branched style of polysaccharides, both in amylopectin, known as starch, found in plant cells, or glycogen, found in animal cells. Both have the same energy storage function. They're usually tightly packed in certain types of cells. In our case, they're mostly stuffed in our liver and stuffed into the different types of muscle cells, areas that need a quick energy store readily available. So you often see most of the long chains going to be linked by alpha 1,4 linkages, so named by the carbon 1 and 4 with each adjacent glucose. But occasionally form branches where there will be alpha chains from the 1,6 carbon bonds, as you see here. So the amylopectin and glycogen have very similar structural positions here. You just simply store it in different types of cells. So again, starch found in plants, glycogen found in animal cells. When you see unbranched linkages with different types of links, in this case in cellulose, this is a unbranched link. These are going to be different types of bonds. These are known as beta 1,4 bonds. These are indigestible by humans. This was known as your insoluble fiber. That's important for our diet. So this helps push things along through our gastrointestinal tract and increase the motility that we need to keep things moving through the GI tract. The fourth one that's not shown here is known as chitin, C-H-I-T-I-N. You find this in insects and arthropods. This is a hard, leathery substance, also a beta bond, which is going to be also undigestible by humans. But it's going to be linked by different types of polymers here. In this case, not glucose, but it's going to be an N-acetylglucosamine, which is basically an amide derivative of glucose. Now, these are all your polysaccharides. Now, oligosaccharides are shorter chains, and these have a different style of function. These can be tied to either proteins and or lipids, which we'll get into when we get into cell membranes. These often will be used as either tags for different types of cells, so the individual organism's body can identify whether it's a foreign organism or whether it's a part of the actual body itself. It also adds in the formation of glucosal layers or mucosal layers to allow sliding of the different types of cells on each other to allow certain types of motility. But very simply, they often act as markers for cell recognition and also for linkages between adjacent cells. So in this case, if we look at skin cells, you want them to be very tightly woven together so they don't break very easily. So the oligosaccharides, besides being a very good energy store, also helps us add the additional function of either markers or some type of fluidity between cells or some of anchoring or attachment. Next set of macromolecules are lipids. Now, lipids have three different functions in the cell. One, they're major energy storage. Second, they are very important for the structure of the plasma membrane and other membrane-bound organelles. So they help with the permeability and also the fluidity of the actual membrane itself by either using phospholipids and or using cholesterol. Now, cholesterol has to be in a certain amount. Otherwise, if there's not enough cholesterol, then the plasma membrane can fall apart. If there's too much, then it doesn't become fluid enough and actually can solidify and not be permeable at all. The third thing is our basis for our steroid hormones, so it's very important for cell recognition, and also very important for cell signaling, especially with the endocrine system. Now, when we talk about lipids, the most basic structure is what's known as the fatty acid chain. So very simply, we have fatty acids which will be either in the saturated, monounsaturated, or polyunsaturated forms. So if you look at palmitate and stearate, the first and second ones here, notice this has a long carbon chain, and this is going to be fully saturated, meaning that every carbon is going to be bonded by at least 4 other different molecules. In this case, this is going to be at least adjacent to the other carbons, as well as 2 hydrogens, except for the last carbon, which has 3 hydrogens at the tail. Those are known as saturated fatty acids. You can have a monounsaturated fatty acid when there's a kink or a double bond, as you see in oleate over on the right here, where the 2 carbons almost dead center of the fatty acid chain causes a kink in this particular area. Now, if there's more than one of those double bonds in the fatty acid chain, it's known as polyunsaturated chains. Now, what happens is on most of the different types of lipids, this is our basic building block. They'll be attached either in sets of 2 or sets of 3. We look at phospholipids or look at triglycerides in the next slide. But any combination of these chains of fatty acids can directly influence the structure and also therefore the function of that particular lipid. Now, our most efficient form of any storage is known as a triacylglycerol, otherwise known as a triglyceride, or even commonly known as fat. So in the previous slide, we saw different fatty acid chains that are going to be linked to a carboxyl group. In this case, we're looking at 3 fatty acid chains linked to a glycerol. And as you notice here, the fatty acids could be in almost any type of situation. You could have a palmate or stearate or even oleate. In this one, it just shows 3 fatty acid chains of palmitate. These are all going to be saturated, so this can actually layer upon layer very easily in fluid droplets found within the cytoplasm. If you look at most animal cells or even in some plant cells, you'll find a lot of fatty acids or a lot of triglycerides will be packed into adipocytes, which are known as fat cells. So very simply, the fat droplets forms a big reservoir of energy storage, which very simply takes over almost the entirety of that particular adipose cell, where the plasma membrane, whereas the nucleus and the rest of the organelles tend to get pushed out to the periphery. And it looks like almost like a diamond ring kind of thing if you look at it under a microscope. Mainly because the triglycerides are not soluble in water, so they tend to congregate and very simply form the fluid acid droplets. Very similar when you put oil in water at home if you're boiling pasta or whatever else. Now, while triglycerides have a glycerol and 3 fatty acid chains, when we look at phospholipids, their structure is a little bit different. The structure is very simply 2 fatty acids joined to a polar head group linked from a glycerol. So very simply, we have 5 of the phospholipids known as glycerol phospholipids. So we see phosphatidic acid, phosphatidylethylamine, phosphatidylcholine, phosphatidylserine, and phosphatidyllinositol are all going to be glycerol phospholipids because they have that glycerol branch off of the fatty acid chains. The one odd one here is called sphingomyelin, which is going to be a choline head that's going to be attached from the serine instead of glycerol. They all have the same basic function, however. They are going to have both a hydrophobic and hydrophilic ends. So the fatty acids, as we mentioned before, these are hydrophobic. So if you remember from our plasma membranes, we have the bilayers. So the fatty acids tend to be hydrophobic, so they're going to be towards the interior of the phospholipid bimembrane. Whereas the hydrophilic ends, the phosphate and glycerol ends, are going to be more facing towards the aqueous solution of either the cytoplasm or the interstitial fluid on either side of the plasma membrane. So very simply, the phospholipids are going to be different from the triglycerides in that they're amphipathic, which means they're both hydrophilic and hydrophobic. They have a hydrophilic end and a hydrophobic end. So they're part water soluble and part water insoluble. So these will automatically orient themselves. In fact, that's one of the things that we looked at. We looked at the RNA world as far as evolution of the cell. If you drop phospholipids into any kind of aqueous solution, they will immediately start to form next to each other and form in that phospholipid bilayer. And then, of course, if some RNA was encased therein, it was protected very simply by the surrounding environment. So the chemical nature of the phospholipids, especially glycerophospholipids, is very integral to how the cell membranes form and how they protect or how they differentiate from the inner portion of the cell versus everything outside of it. Now, similar structure to phospholipids, more so to the sphingomyelin, you'll find different types of what we call glycolipids. These are basically fatty acid chains, 2 fatty acid chains that are linked to a serine that also has a glucose or a carbohydrate tag. So oftentimes these will allow like a slippery nature to the outside of the plasma membrane to allow certain types of cells to slide one another. In particular, think of muscle cells as we go to move muscle back and forth. It allows the muscles to slide upon one another to allow proper motion. It's also important to note that the glucose tag here for the glycolipid can also be a signal to show whether certain types of cells are foreign or whether they're part of the actual organism or not. Now, one thing that's not shown here too, you can also have what's known as a lipoprotein, which has different type of tag. It's also very integral to different types of transport of different types of things throughout, in our case, through our own blood, trying to transport different types of lipids back and forth. Since lipids just dumped into the bloodstream will simply just kind of clog up the area and not really be transferred very easily. These are also amphipathic, so they do have the glucose and the serine heads being very hydrophilic, whereas the fatty acid chains can be hydrophobic. Now, the fatty acid chains shown here, which I did not talk about with the phospholipids before, they can be either saturated or unsaturated. Most oftentimes they are unsaturated, which allows a little more fluidity at room temperatures. The other types of lipids we'll talk about here include the cholesterols, which totally different structure. They are structured as 4 carbon rings with a hydroxyl group at one end. So they tend to be mostly hydrophobic, but with the hydroxyl end attached to the end of the one cholesterol, it becomes very weakly hydrophilic. So in this case, cholesterol itself can also be amphipathic, although somewhat weakly. And here we see the 4 carbon ring of cholesterol, and cholesterol is going to be the precursor for almost all of our different types of hormones, and also a handful of some vitamins as well. So the 4 carbon ring is going to involve 3 hexoses and 1 pentose sugar, And of course, you see the hydroxyl group at the end of the cholesterol above through here. So with different variations from the cholesterol, you can get other types of hormones such as testosterone, estradiol, as all the other fat-soluble vitamins as well. For sake of ease, most of the hydrogen bonds are actually taken off of here, except for the end of the pentose ring for the cholesterol, testosterone, estradiol. But again, this is going to be one of the major signaling sources both in and out of each individual cell, depending on the organism, to either turn certain sequences off or to activate certain types of processes into and out of the cell. And here's the next set of macromolecules, nucleic acids, which comprise our DNA and RNA. So the purines, which are going to be adenine and guanine, are found in both RNA and DNA. You notice there's going to be two sugars here, one a pentose and one a hexose sugar, followed by the pyrimidines, which include cytosine, thymine, and uracil. Notice these are only going to be hexose sugars, just one ring sugar. So the way you remember this very simply, if you're looking at the adenine and guanine, since it's going to be two ring sugar in the purines, use the acronym CUT, C-U-T, to remember cytosine, thymine, and uracil as your pyrimidines, as you're cutting away a 5 ring sugar away from hexose to leave yourself with a hexose sugar there. I won't be testing you on the actual configuration here, just so we know which ones are used in RNA versus DNA, and this should be hammered into you already by now, but uracil is only found with RNA. Its corresponding pyrimidine is thymine in DNA. The sugars are going to be ribose, found in ribonucleic acid. It has a hydroxyl group, which is going to be different from the deoxyribose found in deoxyribose nucleic acid, which simply has the oxygen taken away for the deoxyribose sugar. When you attach one of the purines or pyrimidines to a sugar itself, it is known as a nucleoside. That's your basic nucleic acid. When you add it to a phosphate group, that gives you a nucleotide. And remember from DNA in particular, the nucleotide backbone forms the helix of DNA, as the phosphates will basically adhere to the next adjoining sugar. The nucleic acids will only adjoin to each other through hydrogen bonds. So very simply, the adenine will either pair with thymine or uracil, if we're talking about DNA, RNA respectively, and the cytosine will pair with the guanine. It's necessary to have only one purine and one pyrimidine just to establish, especially in DNA, a uniform diameter in the DNA double helix. And just as we saw with the polymerization of the polysaccharides, we all see the same sequence with the polymerization of nucleotides. So in this case, we're using what's known as phosphodiester bonds, which were binding phosphates to sugars in sequential form. Now, to make this more of a energetically favorable reaction, very simple, what we do is we have one particular nucleotide shown at the top there, added to a highly energetic triphosphate grouped nucleotide here. When they actually form the covalent bond of the phosphodiester bond, it will release the pyrophosphate in a more energy favorable reaction, and it will form that linkage from the 5' to 3' end. So we see this in both the DNA and RNA. We're always going to see it polymerize from the 5' onto the 3' ends. Now, just like when we see saccharides as either the oligosaccharide form or the polysaccharide form, we see the same thing with nucleotides. So we see small strands of nucleotides known as oligonucleotides. If you remember back from Biology 1, those are your Okazaki fragments. Those are small bands of nucleic acids or nucleic bases that come together to fill in fragments as the DNA replicates. Now, if you look at the DNA helix altogether, that is known as a polynucleotide. So polynucleotides in the DNA sequences could be millions of nucleotides long, whereas RNA can be around about 1,000 nucleotides long. Now, this will always bind with a purine to a pyrimidine. In this case, you see cytosine and guanine paired together, and we'll have 3 hydrogen bonds paired between them if you look at the inset on the diagram through here. And conversely, the thymine and adenine will have 2 hydrogen bonds that will bond together. Now, as I mentioned before, having one pyrimidine and one purine at each level of the base pairings here allows the uniformity of the DNA sequence as far as diameter goes. So it keeps the double helix in a very static structure. So you see so many turns within 3.4 nanometers, see 2 turns there, and then 1 turn within 0.34 nanometers at the next 3 sets of bases. So this gives you the double helix of the DNA structure, And again, it's going to follow the phosphodiesterase backbone, the phosphate and sugar backbone, both from the 5' and 3' end. Now, normally RNA forms a single strand. So in the case of messenger RNA, it's just simply no folding. It's just a single strand of a long polynucleotide chain. But oftentimes in different types of RNA, such as transfer RNA or ribosomal RNA, you will get folding based upon the hydrogen bonding between certain pairs of nucleotide bases. So in this case, we see the cytosine and guanine just as we do with DNA. Again, 3 hydrogen bonds between them. And again, you follow the sequence of the 5' and 3' ends back and forth, going in opposing or antiparallel directions. The main difference being is uracil is involved here, but again, it takes the place of thymine. It gives you 2 hydrogen bonds in the adenine through here. Now, this is a particular picture of a transfer RNA. This is responsible for pairing up with a designated amino acid to go with the anticodon found on the tRNA, which will bring it to the associate ribosome to start forming the polypeptide chain that we see for proteins. As mentioned in your text, nucleic acids can direct their own replication, which is essential to make sure that whatever DNA sequence is present in the parent cell can be donated properly to the daughter cells. And as we had seen before, under the semi-conservative model, 1 parent strand is donated to each of the daughter strands, and 1 new complementary strand is going to be synthesized to fully complete the DNA sequence. Now, as long as we get the exact copy of the DNA, if you transfer over to the A column here, so we look at RNA transcription and translation. Very simply, the base pairing, again, will pair up very nicely to give us the proper mRNA sequence, to give us the right protein synthesis with the right amino acid sequence. And the classic example is if any one of those base pairs is incorrect, it could either stop a particular amino acid sequence prematurely and therefore have a dysfunctional protein, or alter the function of the protein entirely by changing how the protein will fold and either create a new particular function or decrease the function entirely. So it's very important that these particular base pairs can self-replicate themselves. Otherwise, the types of problems afterward where the amino acid sequence can change can be somewhat catastrophic to the daughter cell. The most diverse group of macromolecules that we have are amino acids. So these will be the monomers that will form the polypeptide chains, which will form into proteins. Now, proteins can have a myriad of different functions, including structural components for cells and tissues, transport of smaller molecules. They can act as hormones for transmitting signals back and forth. Could also provide against infections such as antibodies. Also act as enzymes and also can be used for movement such as actin and myosin filaments found in muscle. Now, the one basic structure for all amino acids are going to include a carbon or alpha carbon atom with an amino end, which will be a positively charged end, a carboxyl end, which is going to be a negatively charged end. So this will be a polar molecule with a hydrogen adjoining the 3rd carbon bond. The only differing portion, which actually will give each particular amino acid its own property, is going to be the side chain or the R group. And out of the 20 amino acids, it is the side chain that will vary quite a bit from amino acid to amino acid. But this is the most basic structure. So again, you have an amino end or amino group on the positive side. You have a carboxyl group that will be on the opposing negative side. These two will join with a polypeptide bond during dehydration synthesis. Here we see the 20 amino acids that form all the proteins that are available throughout life. Now, I won't get into which ones are essential and non-essential. So the essential amino acids are the ones we need to consume as humans into our diet in order to be utilized for different protein structure, whereas the others can be produced by our own bodies. But since we're talking about the whole of biology, all the amino acids here, whether essential or non-essential, will be applicable in this particular course. Now, I do expect you to know which ones are going to be considered non-polar versus polar, which ones are basic, and which ones are going to be acidic. So let's just point out a few of the non-polar amino acids, and to be fair, I don't expect you to remember the actual structure. I do expect you to know the names of the different types of amino acids, as well as which ones will be non-polar, polar, basic, or acidic. Now, for the non-polar amino acids, these, of course, are going to be hydrophobic. So most of the time, you'll find these on the internal portions of most folded proteins because they want to stay away from any kind of aqueous solution. One thing I want to note is glycine and also glutamic acid. They tend to be also used as neurotransmitters, which we'll get into in a later chapter. Now, the nonpolar amino acids, what of note is if there is a change in the amino acid sequence, meaning if a certain code does get switched, and let's say it goes from one nonpolar amino acid to another nonpolar amino acid, then the change should hopefully be minimal, and there may not be much of a change structurally or functionally for that particular amino acid, although there may be some. It may not work 100%, but it'll still work. However, if you swap out a nonpolar to a polar amino acid, that can definitely change the configuration of the, or 3D configuration of a particular protein, which can definitely alter the function. So the polar amino acids will have some type of charge to them, so they tend to be more hydrophilic, so they'll be more so on the outside portion or the outer portion of facing the aqueous solution in the cytoplasm or interstitial fluids. So again, nonpolar amino acids will tend to be more towards the center of a particular protein, whereas the polar amino acids will be more on the outer portion. The basic and acidic amino acids will also be very hydrophilic, as they will donate, in this case, the basic will donate a hydroxyl ion into solution, or the acid will donate its hydrogen. But the basic amino acids here, lysine, arginine, and histidine, again, will be mostly on the outer portion of a folded protein, whereas an acidic amino acid here will donate the hydrogen, also be more hydrophilic, otherwise known as not only aspartic acid, also known as aspartame, or glutamic acid, also known as glutamine. And looking back at my notes, I do apologize. It is actually glycine and glutamate are going to be the two neurotransmitters. They're very simple amino acids here. Now, when we bind amino acids into a polypeptide chain, the process is somewhat similar with one key difference than when we added different monomers of glucose into polysaccharides. So instead of forming a glycosidic bond, we form what's known as a peptide bond. So the peptide bond is also going to be a dehydration synthesis reaction. So you're going to basically adjoin two amino acids, one at the amino end, also known as the N terminus, that's the positive end, to the carboxyl end, also known as the C terminus, of the next adjacent amino acid by releasing water and adjoining the two in a peptide bond. It's a covalent bond between both amino acids. Now, the one key difference, however, is this is going to occur on the ribosomes found either free-floating in the cytoplasm or on the endoplasmic reticulum. Now, this also is going to be a somewhat unfavorable reaction as far as energy expenditure goes, so it needs to be coupled with another reaction, in this case, donating ATP as well as a GTP to each to actually provide the peptide bond. So very soon what happens is when the different tRNA brings the corresponding amino acid together on the surface of the ribosome will actually catalyze the ATP and actually use the energy of the broken bond of the last phosphate, in some cases two phosphates, to basically convert the amino acyl into amino acyl AMP, whereas the transfer RNA will come and actually bind the amino acyl into tRNA, and then very simply using the phosphate from the GTP will solidify the peptide bond from the N terminus from the C terminus, and therefore to keep adjoining the growing polypeptide chain until it hits the stop codon, and then it will fold itself either in the cytoplasm or in the endoplasmic reticulum. Back in the 50s, there was a biologist by the name of Frederick Sanger that was able to determine the actual structure of a protein. In this case, insulin, known as a hormone, was comprised of two distinct amino acid chains, and they were formed together by what's known as disulfide bonds. So the reactions between two areas of the amino acids that involve cysteine, which will basically form the two sulfurs together and form the disulfide bond, which is a very strong interaction between the two amino acid chains. He was able to determine that if there was any disruption of that particular sequence, then insulin would not be able to work properly. So not only with insulin do we see the interaction between amino acid chains determine the structure of a particular protein, we also see different types of foldings into different configurations. So we start out with what's known as a primary structure, and then into a secondary structure, which we'll get into in a different chapter, which talks about alpha chains and beta plate sheets. But most proteins are going to fold into a 3D or three-dimensional orientation. Oftentimes we'll join with other 3D orientations to form a quaternary structure. But we can actually disrupt the protein folding by doing what's called denaturing a protein. Denaturing a protein can often happen through either heat, changes in pH, or mechanical disruption. But oftentimes these types of denaturation is reversible. One of the things that you can demonstrate at home very easily is the egg white or the protein found in egg whites known as albumin. In liquid form, in room temperature form, they stay liquid. Once you put them under heat, then it denatures the proteins and creates a solid white substance. So right then there is very difficult to reorient that into a renatured phase. However, different types of proteins, especially some found within certain organisms, once they're put back into different favorable conditions, they can actually revert back into its original form. There's a scientist that we'll talk about on the next slide that actually tested this. So he determined very simply that it was without any other cellular functions available. The amino acid chain itself is what determined the actual folding of a particular protein, and therefore also determined that particular function of that protein. So if we disrupt the actual 3D folding, we change the form of it, it changes the function. If we're able to let it reform under certain conditions, it can go back to doing what it normally does under normal activity. Here is biologist Christian Alfredson. He worked with his colleagues Michael Sela and Frederick White Jr. back in 1957. So he basically treated ribonuclease to a certain type of treatment to denature the particular three-dimensional configuration of the protein. So he subjected it to different types of treatments to break down the hydrogen bonds, the disulfide bonds, and reduce them down to different types of sulfohydryl groups. So what this did very simply was it broke down the three-dimensional structure, and they tested the enzymatic activity of the nature of protein. As you would imagine, the enzymatic protein went down to over 0. And then once they were actually able to put it back under a buffer and able to add the presence of oxygen, they noticed that the disulfide bonds had reformed, the hydrogen bonds reformed as well. And they noticed too that the enzymatic reaction of ribonuclease started to go back up. So what they were able to determine very simply was as long as the amino acid chain was intact and simply the different types of bonds were broken, the hydrogen bonds and disulfide bonds, and the amino acid sequence stayed intact, it would then, over time, it would actually reform its particular three-dimensional sequence in exactly the same way it was before. So they noticed too the enzymatic activity will basically reform after a certain amount of time. So they termed this as thermodynamic hypothesis of protein folding. That is very simply that three-dimensional structure is basically inherent to the protein or the amino acid sequence itself, and is basically in its most stable state under physiological conditions. We'll see in the next chart a plot graph that shows very simply how many sulfide groups were still left over after denaturation. We give an idea of the effect of the efficacy of that particular enzyme. See here on the plot graph with the low amount of sulfohydryl groups that are in the solution, meaning that the proteins are still in its fully configured form, the activity stays relatively high. We see an inverse relationship, though. We start seeing the increase in sulfohydryl groups, which is more evidence that the protein is becoming denatured. We see naturally a decrease in activity. So again, it's a direct relationship between the form versus function. If we mess around with the form of the particular enzyme, it'll screw around with the function very easily. But fortunately, for this particular type of enzyme, it was easily reverted back to its renatured position, and therefore the activity was able to get back up to optimal conditions. Another example of the three-dimensional structure of a protein is myoglobin, shown here. So this is a oxygen-carrying protein found in mussels. So this is the fluid that you find after eating a steak or any kind of red meat. It's not blood, but it's actually myoglobin found in the fluid therein. So very simply, before the latest techniques have been out, there's now a computerized system through Google, or actually through one of their sister corporations in 2021, called AlphaFold 2, which actually is a program that helps determine a folded structure of a protein just based on the amino acid, which has shown some promising results as trying to predict how a certain protein should be folded. So this gives a great idea, or scientists a great idea actually, trying to figure out what kind of proteins they can actually form for different types of needs out there in the real world. Now, before that, though, back in the 50s, there was a man by the name of John Kendrew using what's known as X-ray crystallography, which you may have heard about back from Biology 1, where they would shine X-rays through the particular structure and see what kind of diffraction pattern they would have. And depending on what type of atoms that they knew had made up that particular molecule, they can predict what type of structure it would normally have for the particular type of protein. So he actually used myoglobin as his particular template, which is based of about 153 amino acids. So since using X-ray crystallography with relative accuracy, they were able to determine close to about 200,000 different types of proteins. There's about 180,000 out there now, but it's still growing. but they're able to see that very simply that knowing that the amino acid structure, knowing the DNA template, will give you an idea what the mRNA as well as the amino acid sequence will be, so you can predict what the formation will be very easily of a three-dimensional protein. But as I mentioned before, the AlphaFold 2 is a product that is still in development, although it's showing very promising results. Seem to be approaching almost the same accuracy as X-ray crystallography, and if not surpassing it in the near future. So let's bridge the gap between the primary structure of the amino acid up to the secondary, tertiary, and also the quaternary structures of proteins. So I mentioned briefly a few slides back about the secondary and the tertiary structures. So secondary structure are going to include what's known as alpha helices, shown on the top here, or beta-plated sheets. So the alpha helices is very similar to DNA, although not quite the same. This is basically when the amino acid sequence will coil in on itself and sometimes kink or turn due to hydrogen bonding between different areas, between hydrogens, between the oxygens, and often other groups found in the alpha helix. Conversely, the beta-plated sheets shown on the bottom here, those when you see flat sheets of the amino acid chain, either running parallel or antiparallel, depending on where the kink of the alpha helix may be, will also form hydrogen bonds between the hydrogens and the adjoining oxygens. Next, from the secondary structure, we're looking at tertiary or 3D structures. So we see two enzymes here, ribonuclease and beta-glucuronidase. Say that 5 times fast. Beta-glucuronidase, each with its own distinct number of domains. So very similar, we're looking at a ribbon model of both enzymes here. On the left, you see the alpha helices shown as coils, and you see the beta-plated sheets as long, broad arrows. And adjoining each structure there are loop regions. This is where the proteins will fold in on themselves, basically keeping the hydrophobic portions of the particular amino acid sequence more internal and away from the aqueous solution, whereas all the hydrophilic areas, as well as different amino acid groups and carboxyl groups, more on the outside of the particular part of the enzyme. So the ribonuclease here has one domain, whereas the beta-glucuronidase very simply has 3 domains. And the way you can tell this very simply is if you look at the sequence of the alpha helices and the beta-plated sheets, they'll repeat in so many forms. Now, I won't ask you very simply to identify how many domains. I'll tell you in a particular quiz or test question how many domains a particular protein will have, and you'll answer questions related to the 3D structure of that particular enzyme. Now, some proteins, and not all proteins, will reach this particular structure, but some will form into what's known as a quaternary structure. So this is basically a composite of a number of different tertiary structures together. So this is the protein hemoglobin, which we talked about myoglobin before, which only has a single domain. We're looking at hemoglobin, it has 4 domains here. So we're looking at 4 different subsections of the heme group with the beta chains and the alpha helices. So very simply, what you're looking at is 4 different active sites for the heme group. So for the binding of the oxygen onto the iron, as well as the rest of the active site of each particular section of the enzyme. So again, not every protein will reach the quaternary structure here, but hemoglobin is a perfect example of tertiary structures coming together to form the 4D or quaternary structure here. And finally, we'll close out something that's relatively new in the study of biology, talking about intrinsically disordered proteins and regions. So these, if you look at the comparison of the IDP, known as intrinsically disordered protein, versus the IDRs, which is intrinsically disordered regions, they differ greatly from the structured protein. As we mentioned before in the last couple of slides, a structured protein is dependent on the amino acid sequence, and whether we have hydrophobic or hydrophilic parts of the amino acid chain will determine how well the protein will fold in of itself and actually have a functional protein. By design, we actually have different types of proteins known as IDPs and IDRs, which are somewhat flexible. They are transient between different types of positions, so they aid in different types of functions in and throughout the cell. So very simply, the flexible nature allows them to adapt their structure to bind different proteins and form different protein complexes. So very simply, they work with RNA synthesis, they work with RNA processing, what's known as transcriptional regulation, so how often or how much of a particular mRNA strand is going to be transcribed from DNA. Also with nuclear substructures, but we'll talk about this a little more so in both chapters 7 and 10. So all I ask very simply is just be aware of the IDPs and IDRs and how they are going to be a little different structurally than a standard structural protein. Again, these tend to be a little more fluid, to enable them to be involved in a multitude of different reactions.